Okay, so welcome back to this next video in which we are discussing Parkinson's disease and anti-Parkinson drugs. Okay, right, so we have now seen the direct and indirect pathways, okay, and we've seen how the direct pathway is this pathway by which a certain area of the secondary motor cortex activates its permissive signal. Okay, and this isn't the direct pathway. I'll get the direct pathway back. Okay, so here's the direct pathway here. Okay, so this is the pathway by which this area of secondary motor cortex, which has this motor plan which is actually supposed to be initiated, how this portion of secondary motor cortex is going to activate the permission signal coming from the ventral anterior and ventrolateral thalamic nuclei to itself. Okay, right. The indirect pathway then, and I'll just finish off the title, indirect pathway, okay, is the pathway by which this portion of secondary motor cortex, which has the motor plan which is actually supposed to be initiated, how it's going to make sure that the rest of the secondary motor cortex surrounding this doesn't get permission signals coming to it, okay, from the ventroanterior or ventrolateral thalamic nuclei. Okay, so by activating the indirect pathway, it makes sure that the permissive signals coming from areas of the ventral anterior and ventrolateral nucleus to these surrounding areas of secondary motor cortex, that those are turned off, basically. Okay, right. So that then is our model for how the basal ganglia work to control which motor plans are actually going to be sent to the primary motor cortex and initiated, okay, and therefore they control this gate for voluntary movement, basically, okay, and that's why they're often called the gatekeeper, basically. Okay, so what we now want to see is the role of the substantia nigra pars compactor in all of this, because we haven't talked about the substantia nigra pars compactor and the nigrostriatal dopamine pathway at all so far. Okay, so let's now see the role of that. So basically, the substantia nigra pars compactor gives a tonic level of dopaminergic neurotransmission into the dorsal striatum. Okay, so let's put this here. So if we have our substantia nigra, I'll draw this out here. So we'll have our midbrain here. Okay, and I won't actually draw the nigrostriatal tract in the way that it actually appears anatomically. We'll just have a physiological diagram here. Okay, so here's our midbrain. Here is our substantia nigra. And now the portion that we are interested in is the substantia nigra pars compactor. Okay, so the SNCP here. Oh, sorry, SNPC rather. Okay, in yellow here. Okay, now the neurons in this are going to be sending their axons into the dorsal striatum. So I might actually draw this from the side now. So here is the putamen viewed from the left-hand side, and then we'll have the caudate nucleus here. And basically, the substantia nigra pars compactor delivers a tonic amount of dopamine into the dorsal striatum. So this is the dorsal striatum here. Okay, so what I mean by that is that these neurons are firing all the time, okay, and releasing a tonic amount of dopamine into the dorsal striatum here. Okay, so it is not the case that the substantia nigra pars compactor is only going to be activated when you want to make a voluntary movement. No, it's activated all the time, okay, and we don't think that it's involved in actually initiating specific movements. Instead, what we think the substantia nigra pars compactor does is it determines how easy it is to make voluntary movements. Okay, so you know, sometimes you can move your body very, very easily, and then other times, maybe when you're ill, it feels like a much greater effort to move around. Okay, well, this may represent, well, the neural correlate of this may be that the substantia nigra pars compactor and how much dopamine it's releasing into the dorsal striatum here. So let me elaborate on this a little bit more. So we'll colour in the dopaminergic neuron here. in red. Now, in order to understand this more, we need to discuss the type of synapses that dopaminergic neurons are going to form with the neurons in the dorsal striatum. Okay, and they're going to synapse onto both these GABAergic neurons of the indirect pathway and also the GABAergic neurons of the direct pathway. Okay, but they're going to have very different effects on both of them. Right. So let me tell you a bit more about the neurons in the dorsal striatum. Okay, so the neurons of the dorsal striatum have a special name. Okay, they are known as medium spiny neurons, or for short they're called MSNs. So medium spiny neurons, or MSNs. Okay, right, and this is because they're going to be covered in dendritic spines. So let me show you a picture of one of these medium spiny neurons. Okay, so they often have these sort of triangular shaped nuclei, and then they'll have dendrites coming off like so. So here's a dendrite, here's a dendrite, and I think we should put a dendritic spine. Okay, so in case you don't know what a dendritic spine is, the dendrites of neurons are covered in these little processes that look like this, little mushrooms coming off the dendrites. Okay, and these are also known as dendritic spines, and these are the sites of attachment of axon terminals of other neurons. So this is a dendritic spine. And basically, the dendrites of these medium spiny neurons are going to be absolutely covered in dendritic spines. I'm just too lazy to draw more than one, basically, but in reality, the dendrites are going to be covered in these dendritic spines. Okay. So here is the nucleus of this medium spiny neuron here. This is the axon down here. Okay, then we've got the dendrites, and I've drawn 3 dendrites here, and these dendrites will all be covered in dendritic spines. Okay, so let's colour in the different portions here. So we'll have the axon in turquoise here. Okay, we'll have the dendritic spine then here in blue. Okay, that's the other crucial bit here. So, here is one of these medium spiny neurons. Now, some of these medium spiny neurons will be of the direct pathway, and some of them will be of the indirect pathway. Now, both medium spiny neurons of the direct and indirect pathway are going to be GABAergic. So, right at the end of their axon, they'll have an axon terminal, which will be releasing the neurotransmitter GABA. Okay. In addition, both types of medium spiny neuron, both the ones of the direct pathway and the indirect pathway, are receiving activatory signals from the secondary motor cortex up here. Okay, so what I'm going to now draw is a cortical neuron coming down and synapsing on one of these dendritic spines here. Okay, so this is a cortical excitatory neuron. Okay, so it will be releasing glutamate into that synapse and exciting the dendritic spine. Okay, and what will happen is when the glutamate acts on its glutamate receptors, those glutamate receptors will be ligand-gated ion channels, and they'll open and allow in sodium ions into the cell. Okay, and this will be happening at all the different dendritic spines all over the place. And then at the start of the axon, there is a special region known as the axon hillock. And what will happen is all of these currents will be coming together from all of the different dendritic spines. Okay, and if those currents, those sodium currents, are substantial enough to trigger an action potential, the action potential will start from the axon hillock and then propagate down the axon. Okay, so the axon hillock is the starting point of the actual action potential. Before that, it's just ionic currents coming in from the dendritic spines, basically. Okay, so that's how these cortical neurons are going to be exciting our medium spiny neurons of the dorsal striatum. Okay, now where are the dopaminergic neurons going to be synapsing onto these medium spiny neurons? Well, generally the dopaminergic neurons synapse onto the necks of the dendritic spines, and I'm sorry it looks a little bit odd. The neck is tiny of this dendritic spine, so that's why I've had to show it like that. So you see the dendritic spine is in this sort of mushroom shape, and the cortical neuron is synapsing onto the sort of head of the mushroom. But there's that neck of the mushroom as well, known as the neck of the dendritic spine. Okay, and it's the neck onto which the dopaminergic neurons are going to synapse, and they're going to be releasing dopamine onto the necks of the dendritic spines. Now, what the dopamine neurons then do is they moderate how easily the cortical neuron can activate the dendritic spine. They moderate how much sodium current you're going to get coming in through the dendritic spine every time the cortical neuron releases glutamate onto the dendritic spine. Okay, so these are moderatory, basically. So the cortical neuron releases glutamate, which then activates a sodium current. But how big is the sodium current? Well, that's going to be controlled by this dopaminergic neuron here. Okay, and basically, dopamine coming onto the neurons of the direct pathway. Okay, so on the direct pathway neurons, you're going to increase the amount of sodium coming in into these dendritic spines every time the cortical neuron releases glutamate on. Okay, so dopamine acting on the direct pathway is going to cause sodium current to go up. Okay, so it's going to make it easier for the cortical neurons to activate direct pathway medium spiny neurons, and on indirect pathway medium spiny neurons, dopamine is going to make it more difficult for the cortical neurons to activate them. Okay, so the sodium currents that are going to come in every time the cortical neuron releases glutamate onto the dendritic spine is going to be reduced after you've released dopamine onto the neck of a dendritic spine of an indirect neuron. Now you might ask, well, why is that? Well, they're going to have different types of dopamine receptors on them. Okay, so let's just have a little bit of a talk about dopamine neurons then. Okay, so dopamine receptors rather than dopamine neurons. Okay, so dopamine receptors then. Okay, dopamine receptors are all in the massive great family of G protein coupled receptors. Okay, so they're all within the massive great set of GPCRs. So this is a math symbol. This means are contained within the set of GPCRs, or G protein coupled receptors. Now, in humans alone, we found, I think it's around over 800 GPCRs now. Okay, so there are a huge number of different GPCRs. Now, 5 of the known GPCRs in humans are dopamine receptors. They respond to dopamine. Okay, so these 5 different GPCRs for dopamine then are very sensibly called D1, D2, D3, D4, and D5. And they all have the characteristic structure of G protein coupled receptors. Otherwise they wouldn't be G protein coupled receptors. So what are the characteristic features of G protein coupled receptors? Well, there is one that rules over all others, which is that you have 7 membrane spanning alpha helices. So all G protein coupled receptors are a single polypeptide. Now that distinguishes them from, for instance, the ligand-gated ion channels, because the ligand-gated ion channels are not a single polypeptide. Instead, they're multiple polypeptides stuck together to make the full channel. Okay, but G protein coupled receptors, they truly are just a single polypeptide, like so. So this line represents the polypeptide. It represents the polymer of amino acids. Okay, and you begin with the amino terminus extracellularly. Okay, so this is the extracellular side of the membrane here, the ECF standing for extracellular fluid. Okay, then you've got the cell membrane here, which is shown as the phospholipid bilayer, so I've shown both the outer leaflet and the inner leaflet. And then the amino terminus of this polypeptide starts off extracellularly. Then you have 7 membrane spanning alpha helices across the cell membrane, finishing up with the C terminus of the polypeptide intracellularly. Okay, right. Now all 5 of these dopamine receptors then have the same membrane spanning alpha helice. They have the same membrane spanning topology. They all have 7 membrane spanning alpha helices. Okay, right. Now the dopamine receptors are grouped into 2 groups. Okay, D2, D3, and D4 are all put into a group, and this group is the D2 type dopamine receptors. Okay, and that leaves D1 and D5 to form another group, which is known as the D1 type dopamine receptors. Okay, so these 2 here, so let's underline them. So D1, and D5 are grouped together. Sorry, D1 and D5 are grouped together as D1 type. Okay. D2, D3, D4 are all grouped together as D2 type dopamine receptors. Okay, now what's the difference then between D1 type and D2 type dopamine receptors? Well, the D1 type, these are going to be activatory. Okay, those are going to stimulate GS heterotrimeric G proteins, the stimulatory heterotrimeric G proteins, and the D2 type dopamine receptors are going to stimulate GI heterotrimeric G proteins, the inhibitory heterotrimeric G protein. Okay, so basically on direct pathway medium spiny neurons, you are going to have D1 receptors. Okay, and on the indirect pathway medium spiny neurons, you are going to have D2 receptors. Okay, and this is how dopamine being released into these synapses within the dorsal striatum is going to have these two very different effects on direct pathway medium spiny neurons and indirect pathway medium spiny neurons. Okay, now let's start to put all of this together then. So, the substantia nigra pars compacta is tonically releasing dopamine into the dorsal striatum. Okay, so it just releases dopamine happily away all the time. Okay, now the dopamine acting on direct pathway medium spiny neurons is acting to sensitize these dendritic spines so that every time a cortical neuron releases glutamate on to the dendritic spine, the amount of sodium current that you're going to be allowing in, which is pro-action potential, okay, it's going to excite the neuron into firing an action potential, that current is going to be getting bigger. Okay, so that's what dopamine is doing. It's ensuring that these currents are going to be big in direct pathway medium spiny neurons. Whereas dopamine acting on D2 receptors on indirect pathway medium spiny neurons, that's going to be inhibiting the sensitivity of the dendritic spines. Okay, so every time a cortical neuron fires, that's going to release glutamate on, and now because of the dopamine acting on D2 receptors on the indirect medium spiny neurons, you're going to get less sodium coming in for every glutamate burst that you release onto the dendritic spine. Okay, so dopamine acts to desensitize the indirect pathway medium spiny neurons. So it acts to activate the direct, or make more activatable the direct pathway medium spiny neurons, and make less activatable the indirect spiny neurons. Okay, now let's just put this all together. Remember the direct pathway medium spiny neurons. Those were necessary, and here they are. These are the direct pathway medium spiny neurons here. This one here going to the internal globus pallidus, and this one here going to the substantia nigra pars reticulata. Okay, these are now going to be more activatable. Okay, and they, remember, inhibit the internal globus pallidus and the substantia nigra pars reticulata, or at least certain areas of these two nuclei. Okay, and by inhibiting those certain areas of these nuclei, we then stop those areas inhibiting certain areas of the ventroanterior and ventrolateral thalamic nuclei, and now those areas of the ventroanterior and ventrolateral thalamic nuclei give permission to this area of the secondary motor cortex to fire the motor plan onwards, basically. Right, so by making the direct pathway medium spiny neurons more activatable by the cortex, you are making it more easy to initiate a voluntary movement. Okay, right, now let's talk about the inhibition of the activatability of the indirect pathway medium spiny neurons. Okay, so here is our indirect pathway medium spiny neuron. Okay, this is now having its activatability toned down. Now let's remember what this neuron did. Well, basically, when this area of the secondary motor cortex decided that it needed its motor plan to go forward into the primary motor cortex, it activated certain areas of the caudate putamen, or the dorsal striatum, and that not only activates the direct pathway, but it also activates many indirect pathway neurons, which are then going to inhibit neurons in the external globus pallidus. Okay, these these neurons in the external globus pallidus are now no longer going to inhibit the neurons in a certain area of the subthalamic nucleus, and these neurons in this certain area of the subthalamic nucleus are going to activate neurons in the internal globus pallidus and neurons in the substantia nigra pars reticulata. Okay, and the areas of these two nuclei that you are going to activate are going to be the areas corresponding to the areas of the secondary motor cortex outside of the portion of secondary motor cortex with your desired motor plan. Okay, these ones are then going to fire more, okay, and they're then going to inhibit areas of the ventroanterior and ventrolateral thalamic nuclei more, and these areas of the ventroanterior and ventrolateral thalamic nuclei that are going to have increased inhibition now will be the areas that supply the areas of the secondary motor cortex outside of our area of interest. Okay, and that's how we stop permission going to areas of the secondary motor cortex outside of the area that we actually want to give permission to. Okay, so you're going to dampen down the indirect pathway and you're going to up the direct pathway. So dopamine as a whole just basically says, okay, we'll give permission to as much as possible, basically. Dopamine is the ease of, well, it's a signal that makes it easy to initiate voluntary movements. It makes it easy to initiate movements. It makes giving this permission signal easy. Okay, so to summarize then, if you have very high levels of dopamine in the dorsal striatum, and dopamine levels will change, okay, and they'll dictate how easy it is to make movements, basically. Okay, so if you have very high levels of dopamine in the dorsal striatum, then you're going to get too much movement, basically. Okay, you're going to get hyperkinesia. Okay, it's going to make it too easy to get this permission signal. Okay, so you're going to get permission signals going to areas of secondary motor cortex that really should not get permission signals, and you're going to get motor plans initiated that you never actually wanted to initiate. Okay, and that's called hyperkinesia. Okay, if on the other hand dopamine is too low, okay, then you're not going to be able to initiate the movements that you want to initiate, because the direct pathway is now going to be too activated, okay? Sorry, it's going to be too underactivated. It's going to be too difficult to activate now, because remember, the dopamine sensitizes those dendritic spines on the medium spiny neurons of the direct pathway via D1 receptors. So if dopamine is too low, that's what's going to cause hypokinesia. Okay, right, so now tying this all back now into Parkinson's disease. In Parkinson's disease, what happens is you get this neurodegenerative disease that takes place all over the brain. However, one of the areas that is specifically sensitive to degeneration via alpha-synuclein is the substantia nigra pars compacta. Okay, so the dopaminergic neurons in the substantia nigra pars compacta start to die. Okay, that means that the amount of dopaminergic transmission that you're going to have in the dorsal striatum is going to go down. Now, when it goes down to around 70% of what it should be, then you start developing the hypokinesia, which is the cardinal feature of Parkinson's disease. Okay, so because dopamine is too low, dopamine is this signal that says, yes, let's make movements easy, okay? When dopamine gets very, very low, it means that allowing these movements, allowing motor plans in the secondary motor cortex to get this permission signal that then says, yes, you can go on to the primary motor cortex, that becomes extremely difficult. Okay, and therefore you then develop the hypokinesia. Okay, you get it becomes extremely difficult to initiate voluntary movements. Okay, so that then is our theory of how Parkinson's disease, this neurodegenerative disease, causes that cardinal symptom, which is the hypokinesia, the difficulty in initiating voluntary movements. Okay, right. So what we're now going to turn our attention onto is the anti-Parkinson drugs. Okay, Now, the anti-Parkinson drugs are all going to work to increase dopaminergic transmission in the dorsal striatum. Okay, that is all they are going to do. Okay, so that is why they do not help with the other symptoms of Parkinson's disease. Okay, too little dopamine in the dorsal striatum can explain brilliantly why you get hypokinesia. Okay, but it can't account for why you get some of these other motor symptoms such as tremors. Okay, and it certainly can't explain why you get the cognitive symptoms of Parkinson's disease and the other non-motor symptoms such as depression and dysautonomia as well. Okay, those effects are resulting from degeneration elsewhere, and replacing dopaminergic transmission in the dorsal striatum is not going to fix those. However, replacing dopaminergic transmission in the dorsal striatum is absolutely fantastic at getting rid of the hypokinesia. And initially, when early stage Parkinson's disease patients take the anti-Parkinson drugs, they basically return them almost to normal. Okay, so they are fantastic at getting rid of the hypokinesia. Okay, right. The other thing that I do need to say is that they are not going to stop the neurodegeneration. We have no drugs that can stop the neurodegeneration of Parkinson's disease. So it continues to progress, and eventually, often, the progression of the disease beats even the anti-Parkinson drugs, and eventually the anti-Parkinson drugs generally stop working. Okay, but they do work for a good period of time. Okay, right. So, to discuss the anti-Parkinson drugs then, we firstly need to have a very good understanding of how the axon terminals of dopaminergic neurons work, because we need to know a lot about dopaminergic transmission in order to understand these anti-Parkinson drugs. We need to know about how dopamine is synthesized within axon terminals, and we need to know about how it's released from neurons. Okay, right. So firstly, let's start off with a picture then of our dopaminergic axon terminal. Okay, so this is going to represent the axon terminal of our dopaminergic neuron here. Okay, so in the axon terminal of our dopaminergic neuron, which will be somewhere within the dorsal striatum, so it's got its cell body far away in the substantia nigra pars compacta, but it has sent its axon through the nigrostriatal tract, and its axon terminal is now in the dorsal striatum, synapsing onto the neck of some dendritic spine of some medium spiny neuron. Okay, now the dopamine neurotransmitter itself is going to be synthesized within this axon terminal, and that's the first thing that we want to discuss, the synthesis of the neurotransmitter. Okay, right, so it's going to be synthesized from an amino acid, and the amino acid we're going to use to synthesize dopamine is tyrosine. Okay, so let me start off by showing you the structure of tyrosine. Okay, so we'll start off with the core amino acid structure then, and now we are really showing a free amino acid rather than the amino acid residue. Okay, so remember long ago now when we were discussing those mutations in alpha-synuclein, we were drawing amino acids as residues. We were drawing them as though they were in polypeptides. Now we're talking about the true free amino acid. Okay, so here's the amino group, here's the alpha carbon with a hydrogen coming off it, here's the carboxylic acid group down here, and then the R group of a tyrosine amino acid consists of a methylene group, like so, and then you're going to have a benzene ring off the side here. Okay, and I'll draw the benzene ring just in its skeletal form, like so, and then an alcohol group coming off the benzene ring here. Okay, so that's the structure of the amino acid tyrosine then, and this is the starting point for producing the neurotransmitter dopamine. Okay, right, so let's see the first transformation that's going to occur to tyrosine. And it's a nice short pathway. There's only two chemical reactions that are going to occur. Okay, so the first reaction that we're going to do is to add on an alcohol group onto tyrosine. And this is going to be catalyzed by a cytoplasmic enzyme known as tyrosine hydroxylase. Okay. Okay, so this is the enzyme that's going to catalyze this conversion here, tyrosine hydroxylase. Now what are you actually going to do? Well, you're going to stick an alcohol group onto this carbon of the benzene ring here to create the catechol ring, remember. Okay, so tyrosine at the moment does not have a catechol ring in it. It has a benzene ring with an alcohol group on, which is known as a phenol ring. Okay, but it doesn't quite have a catechol ring. So we're going to convert this phenol ring here into a catechol ring. Okay, right. So let's now add this additional alcohol group onto the benzene ring. So here's the amino group once again. Here's the alpha carbon with a hydrogen coming off it. Here's the carboxylic acid group. Okay, then we've got the methylene group here. Okay, then now here is going to be our catechol ring. Okay, so here's our benzene ring, and now we're going to have two alcohol groups coming off the benzene ring, one here and one here. Okay, and this molecule now, this is what's known as DOPA. Okay, so what does DOPA actually stand for? Well, DOPA stands for dihydroxyphenyl alanine. Okay, so dihydroxy, that's the D and the O. So the D is for dihydra, and then the O is for the oxy bit here. Okay, and then it's dihydroxy, and it should all be one word, but I certainly don't have space for it here. Okay, and then the final bit is phenyl alanine. Now, phenyl alanine is another amino acid. Okay, if we wanted to draw phenyl alanine, it would be tyrosine without the alcohol group. Okay, so we can see that what we have here is phenyl alanine with two alcohol groups put onto it, basically. Okay, so that is dihydroxy, which is just another name for an alcohol group, phenyl alanine. Okay, so the PA is for phenyl alanine, so dihydroxyphenyl alanine, or DOPA. Okay, right. So now what we're going to do is we're going to convert the DOPA here into dopamine. Okay, right. So to do this, what we're going to do is we're going to take the carboxylic acid group off the alpha carbon and replace it by hydrogen. Okay, and that will then synthesize us dopamine, and you will hopefully recognize the structure that we have created from the picture that I drew of dopamine when we were discussing the nigrostriatal tract. Okay, so now the carboxylic acid group has gone. Okay, so the alpha carbon now just has two hydrogens coming off it, and then we still have our catechol ring here. Okay, here's the benzene ring with the two alcohol groups coming off. Okay, right. Now, this reaction is going to be catalyzed by an enzyme known as Dopa Decarboxylase. Okay, which makes sense because it is removing the carboxyl group from a DOPA molecule. However, this enzyme also has another name which I'd like to discuss, mainly because it's going to save us time later having a discussion about what L-DOPA means. Okay, so this enzyme also has another name which is commonly used, and the other name which is commonly used is to call it L-aromatic amino acid decarboxylase. Okay, so the decarboxylase bit hasn't changed. Okay, what's changed is that rather than calling it Dopa decarboxylase, we are now calling it L-aromatic amino acid decarboxylase. And for short, this is often abbreviated to LAAD for short, L-A-A-D. Okay, L for L, A for aromatic, A for amino acid, and then D for decarboxylase. Okay, right. So this is just another name for this same enzyme. Now, why is it also called this? Well, we will come back to the L. Okay, I'm going to talk through that now because that's going to be very, very important. Okay, but let's start with the aromatic amino acid. Okay, so in reality, this enzyme doesn't just remove the carboxyl group from DOPA molecules. It has a bunch of other targets as well that it can do this same reaction for. Okay, generally it likes amino acids which have an aromatic group in, which DOPA certainly does. The catechol ring is an aromatic group. Okay, so that's how this name relates to this. It's acknowledging that actually this enzyme that initially when we discovered it we thought just took the carboxyl group off DOPA, it actually takes the carboxyl group of a whole bunch of aromatic amino acids. Okay, now this L, what does this L mean? L-aromatic amino acid. Okay, this is referring to the optical isomer of amino acids that this enzyme likes to actually remove the carboxyl group from. So let me just remind you of the two optical isomers of amino acids then. Okay, right. So, if we draw our basic amino acid structure out now, so we're taking a step back, we're taking a step away from DOPA for a moment, and I just want to explain to you the difference between L amino acids and D amino acids. Okay, I want to make sure everyone understands that. So, here is our core amino acid structure, and now we'll just put the R group as an R there, just to mean it's an arbitrary group that you've got there. Okay, so basically, this carbon here, okay, this has 4 different groups coming off it, and therefore it is what is known as a chiral center, and it results in the amino acid molecule having 2 different forms, basically, and these 2 different forms are known as the 2 different optical isomers of the amino acid. Okay, so let me explain to you why these 2 forms arise. Now, to explain this, we need to go away from drawing 2-dimensional pictures like this and go over to drawing 3-dimensional pictures, because we can't understand this just using the 2-dimensional picture here. We have to use a 3-dimensional picture. Okay, right. So, what I'm going to do is I'm going to imagine putting the alpha carbon here at the center, and I'm going to imagine that I have put it in the plane of the piece of paper. So it's just sitting there in the same plane as the piece of paper. Okay? Right. Now I'm also going to say that this hydrogen atom that is attached to the alpha carbon, I'm also going to have that positioned so that it's in the plane of the piece of paper. Okay, finally, I can also choose one further group that comes off this carbon to also be in the plane of the piece of paper, and I'm going to choose this nitrogen atom of the amino group. So I've aligned the molecule so that all of these atoms within this molecule are sitting in the plane of the piece of paper. Now, the 2 final groups that come off this carbon, which are the R group and the carboxylic acid group, one of them will have to come off at an angle like this. Okay, it will be coming out of the page towards us, and it will be going off down here, kind of. It will be going off in this sort of a direction in which I'm pointing my pen. Okay, the other group is going to have to go into the page away from us at an angle like this. Okay, so it's going to have to go into the page, and it's going to have to go down at an angle like that. Okay, right. So, basically, how do you decide which of these 2 groups goes in each of these directions? Do you have the carboxylic acid group coming out of the page towards us and the R group going into the page away from us, or do you do it the other way around? Do you have the carboxylic acid group going into the page away from us and the R group coming out of the page towards us? Okay, so I'm going to show a bond coming out of the page towards us like so. I'm going to show it by the bond getting thicker towards the end. Okay, and this is pretty conventional notation. A lot of people show bonds coming out of the plane of the piece of paper towards you, like so. Okay, let's say that the R group is going to come out of the plane of the piece of paper towards us. Okay, that means that the carboxylic acid group has to go into the page away from us. Okay, now the way that I show bonds going into the page away from us is by this dashed line, like so. Okay, so the carboxylic acid group will go into the page away from us. Okay, so that is one form of the amino acid. Now the other form is going to be where we have swapped these 2 around. Okay, so let me draw this now. So we're going to leave the alpha carbon, the hydrogen and the amino group completely where they are. Okay, then now what we're going to do is we're going to swap that R group and that carboxylic acid group around. Okay, so now the carboxylic acid group is going to be coming out of the plane of the piece of paper towards us, and the R group is going to be going into the plane of the piece of paper away from us. Now, these are 2 two separate forms of this molecule. Okay, you cannot change this one into this one without performing a chemical reaction. You would have to break this bond, and swap them around. Okay, that's a chemical reaction. So you can't turn this one into this one without performing a chemical reaction. No way of just rotating this molecule around or changing your own position by which you look at this can make this one look like this one. Okay, they are fundamentally different. Okay, they are known as different optical isomers of the amino acid. Okay, now there's another word that people use which just confuses this matter. Okay, and the other word people use to describe two amino acids like this, okay, is to say that they are enantiomers of one another. Now what that means is that they are very special optical isomers of one another. In more complicated optical isomer cases, okay, so if you come up with much more complicated molecules with a huge number of different chiral centers, you can come up with many more than two different optical isomers, okay, and two optical isomers, any two optical isomers that you pick of these more complicated molecules are very unlikely to be enantiomers. But in the case of the amino acids, these two are enantiomers of one another. Now what does that mean? It means that they're actually mirror images of one another. Firstly, it means that they're optical isomers of one another and they're actually mirror images of one another. Okay, so how do you see that they're mirror images of one another? Well, if you imagine putting a mirror here, and imagine what this one would look like in the mirror. Well, it would actually look like this one. Now, unfortunately, I haven't actually drawn it the right way around. But imagine rotating this around 180 degrees. Imagine spinning these bottom groups here. Okay, so imagine spinning this around 180 degrees. The carboxylic acid group will go from being down here at the front to being going away from us into the page and going down here somewhere. Okay, the R group, which is currently over here, okay, so it's on the right hand side and it's going into the page away from us, it will now come around 180 degrees and it will be coming out of the page towards us and it will be coming down here. The amino group, once it's spun around 180 degrees, it will still be in the plane of the piece of paper and it will be going back round there. Now, that's the exact mirror image of this, because look, the carboxylic acid group, if we took the mirror image, it would be going down here. Okay, and indeed, when we spin this around 180 degrees, it would be going down there. Okay, the R group, okay, that will be coming out of the page towards us down here, and indeed, that's where it would be coming if we just spun these three around by 180 degrees. And finally, the amino group would be going away from us. Sorry, it would be going off to the right down here, okay, and in the plane of the piece of paper. Okay, so that one's the mirror image of that one, which is why they're called the two different enantiomers. Okay, now, the two different enantiomers of amino acids are labelled up. This one is called the L amino acid here, okay, or the L enantiomer of the amino acid, and this one is called the D amino acid. Now, how do you remember which one is called the L amino acid and which one is the D amino acid? Well, there's a quite handy little trick. Okay, so what you do is you imagine that you are this hydrogen up here and you are looking down on these three groups and you try and write the word corn, or rather you try and read the word corn. Okay, now how do you read that? Well, you start with the CO from the carboxylic acid group. You then get the R from the R group, and then you want the N from the amino group to read corn. Now, if I was sitting up where the hydrogen is on this L amino acid, and I wanted to read the word corn, what direction would my head turn in? Well, it would start here with the CO, okay, then it would go to the R, and then it would go round to the amino group, so my head would be going in a clockwise direction, okay? So, if to read the word corn, your head has to turn in a clockwise direction when you're sitting up where the hydrogen is, that is the L amino acid, okay, and it stands, if you really want to know, for larvorotatory, okay? If we do the same experiment for the D amino acid, okay, and I sit up here and want to read the word corn, I start here with the CO, okay, then I go round to the R, okay, and then finally round to the amino group for the N. Okay, and what direction has my head gone in this time? Well, it's turned anti-clockwise, I hope you can see. Okay, so if you have to turn your head anti-clockwise to read the word corn when sitting up where the hydrogen is, then that's the D enantiomer, or the D amino acid, and it stands for dextrorotatory. Okay, right, so the amino acids that are used within cells are nearly always the L amino acids. D amino acids are really rare in biology. L amino acids are the thing. They're the in thing. Okay? So usually people do not bother specifying which type of amino acid they are talking about. So for instance, I just talked about tyrosine and didn't specify whether it was D-tyrosine or L-tyrosine. It was just assumed that you understood that this was L-tyrosine, because whenever we work with amino acids, it's always the L enantiomer, because those are the ones which cells actually use. Okay, so this was L-tyrosine, and therefore this was L-DOPA, okay, and this, well, this has lost its chiral center because it no longer has the carbon with 4, oh sorry, you can't see it. Dopamine doesn't have the chiral center because it no longer has the carbon with 4 different groups on it. Okay, so if you actually try drawing this out, you'll realize that there aren't 2 different optical isomers here. Okay, you can, if you try drawing out the L and the D forms of dopamine, you will be able to superimpose them on one another without breaking chemical bonds. Just by rotating it around, you can superimpose them on one another. Okay, right, so that was just an explanation of what an L amino acid is, and that's going to come in very useful later when we discuss the therapeutic drug L-DOPA, which really is just this molecule that I've shown here. Okay, so we'll come back to that. Okay, right, so this is all happening inside the axon terminal. So tyrosine, we start off with, it's being converted into dopamine by these 2 conversions that we've talked about now. Okay, and what's then going to happen is the dopamine is going to be loaded into synaptic vesicles. Okay, so once you've created the dopamine in the axon terminal, you don't just release it away now. You need to store it until an action potential comes along. Okay, so it's going to be stored in synaptic vesicles until an action potential comes along. So this is a special membrane-bound structure within the cytoplasm of the axon terminal known as a synaptic vesicle. Okay, right. So, how do you get the dopamine into the synaptic vesicle? Okay, well, you use secondary active transport. Okay, now what is the secondary active transport second to? Well, it's second to a proton gradient. So I'm firstly going to talk to you about the establishment of a proton gradient across the synaptic vesicle membrane, and then we'll come on to how we're going to use this proton gradient to move dopamine into the synaptic vesicle. Okay, so basically, in the membrane of this synaptic vesicle, you have a protein known as the VATPase. Okay, now what does the V stand for? Well, this stands for vesicular, which makes sense because it's sitting in the membrane of the synaptic vesicle. Okay, so let's colour in this vesicular ATPase then in vivid purple here. So this is the VATPase. Now, what does the VATPase do then? Well, basically it pumps protons from the cytoplasm of the axon terminal, okay, into the lumen of the synaptic vesicle. Okay, and it is an active transporter, and specifically it's what's known as a primary active transporter. Okay, now what does primary active transporter mean? Well, primary active transporter means that you're going to couple this movement of the proton into the synaptic vesicle to some sort of chemical reaction, and the chemical reaction that's usually used by primary active transporters, and which is indeed used in the case of the VATPase, is the hydrolysis of ATP. Okay, right, so hydrolyzing ATP is going to release the energy that's going to be used to move this proton into the synaptic vesicle. Okay, so the synaptic vesicle is covered in these vesicular ATPases, which are continuously pumping protons into the lumen of the synaptic vesicle. Okay, now that means that you have an incredibly high concentration of protons in the lumen of the synaptic vesicle, and this is how we're going to get the dopamine in. Okay, so for the purposes of dopamine transport into the synaptic vesicle, there is a transporter known as VMAT2. Okay, now what does VMAT2 stand for? It stands for the vesicular, okay, once again that makes sense, and then the MA is for monoamine, so M for mono, A for amine, and then it's the vesicular monoamine transporter 2. Okay, so this is going to transport monoamines into the synaptic vesicle, of which dopamine is an example. It has a single amino group here. Okay, there are other monoamines such as serotonin and noradrenaline and adrenaline as well. Okay, there is a VMAT1, okay, as well, but VMAT2 is far more important in axon terminals of neurons. Okay, so it's going to mainly be VMAT2 that we've got within the axon terminal of our neuron here. Okay, so we'll have VMAT2 underlined in green here. Okay, and that's the vesicular monoamine transporter 2. Right, so the vesicular monoamine transporter 2 is what's known as a secondary active transporter. Okay, which means that it's going to transport something up its concentration gradient or up its electrochemical gradient if you're being more fancy and more correct. Okay, but it's not going to get the energy for transporting the dopamine up its electrochemical gradient from a chemical reaction. Instead, it's going to get the energy it needs to pump a dopamine into the lumen of the synaptic vesicle from ionic gradients. Okay, so it's going to use this extremely high proton gradient that we have across the synaptic vesicle membrane. So basically what it's going to do is it's going to allow 2 protons to leave the synaptic vesicle, okay, and go back into the cytoplasm. And when they go back into the cytoplasm, they'll release a lot of energy, and that energy is what's going to be used to pump the dopamine into the synaptic vesicle. Okay, so this is an example of a secondary active transporter, because rather than coupling the movement of the dopamine to a chemical reaction, which is what a primary active transporter does, it couples the movement of dopamine to the movement of another substrate molecule, specifically these protons, okay, and they're going to go in the opposite direction to the dopamine, down their electrochemical gradient. Okay, right, so that's how we get the dopamine into the synaptic vesicle. Now, something else that we need to discuss is the fact that dopamine can also be destroyed within the cytoplasm of the axon terminal, and in fact not all the dopamine which you produce is actually going to end up in synaptic vesicles. Some of it is going to be produced only to be destroyed. Okay, right, so the main enzyme there are 2 enzymes which are going to be involved in destroying dopamine within the axon terminal. The first that I'm going to talk about is what's known as monoamine oxidase. Okay, so let's talk about monoamine oxidase then. So, first thing to say is that there are 2 forms of monoamine oxidase. Okay, there is monoamine oxidase A and there is monoamine oxidase B. And for short, they are abbreviated down to monoamine, so MA, oxidase, and then A for the monoamine oxidase A, and then monoamine oxidase B. Okay, now, of these 2 forms of monoamine oxidase B is the more important one for dopamine. Okay, monoamine oxidase A is very good for serotonin, okay, but for dopamine, it's monoamine oxidase B that's the better one. Okay, now where is monoamine oxidase B located in the cytoplasm of the axon terminals? Well, it's actually attached to the outer membranes of the mitochondria within the axon terminals. So let me draw this here. So let's say that this is the outer membrane of a mitochondrion, and here is the inner membrane of a mitochondrion. So I'll just remind you of the structure of mitochondria. So mitochondria have these 2 membranes. Okay, they have an inner membrane, which is this one here, okay, which invaginates in to form these invaginations known as cristae, okay, and then they have an outer membrane as well, with a space in between these two membranes, okay, known as the intermembrane space. Okay, right. Now, monoamine oxidase enzymes are going to be attached to the outer membrane of mitochondria, okay? So, let me show this. So basically, what they have is a single membrane spanning alpha helix, which is going to cross the outer membrane of the mitochondria, and then you're going to have the C-terminal of the monoamine oxidase enzyme inside the intermembrane space. Then you'll have the main bulk of the enzyme in the cytoplasm, with the amino terminus also in the cytoplasm. So this is the basic structure of both of these monoamine oxidase enzymes, both monoamine oxidase A and monoamine oxidase B. You start with the amino terminus extracellularly, you then have the main enzyme portion cytoplasmically. You start off with the amino group cytoplasmically, so this is the mitochondrion sitting within the cytoplasm. This is facing into the cytoplasm. The main enzyme lump here is in the cytoplasm, and then you have a single membrane spanning alpha helix spanning the outer membrane of the mitochondrion, and then you finish with the C-terminal in the intermembrane space of the mitochondrion. Okay, so associated then with the mitochondria in the axon terminal, we have the monoamine oxidase B enzyme. So let me put this here. So here is a mitochondrion shown here. Okay, and then attached to the mitochondrion, we are now going to have one of these enzymes here. Okay, which is the monoamine oxidase B enzyme. So some of the dopamine that you create is not actually ever going to make it into the synaptic vesicle. Some of it is going to be destroyed by the monoamine oxidase B enzyme, which is attached to the outer membrane of the mitochondrion. Okay, now, there is another enzyme that is also in the cytoplasm of the axon terminal, which is capable of destroying dopamine. Okay, so let me mention that enzyme as well. So the other enzyme besides monoamine oxidase is what's known as catechol-O-methyl-transferase, okay, which for short is usually abbreviated down to COMT. Okay, so C for catechol, O for O, M for methyl, T for transferase. Now, there are two splice variants of catechol-O-methyl-transferase. Now, let me just make sure that everyone is familiar with the concept of splice variants. So basically, for this enzyme, catechol-O-methyl-transferase, there is only one gene, okay? Whereas, in the case of monoamine oxidase, the monoamine oxidase A and the monoamine oxidase B, they were from different genes, okay? So in the case of catechol-O-methyl-transferase, there are going to be two forms, but they're going to be from the same gene, okay? And they're going to be what are known as different splice variants of the same gene. Okay, so let me explain how different splice variants come to be. So, let's talk about the central dogma of biology then. So, let's let these two parallel lines that I'm drawing here now represent a piece of double-stranded DNA. So it doesn't represent the phospholipid bilayer anymore, it represents a piece of double-stranded DNA. Okay, and let's say that this portion that I'm outlining here, okay, and I think I'll even highlight it, let's say that this portion of the double-stranded DNA is some gene. Okay, maybe it's the gene for catechol-O-methyl-transferase. Okay, COMT gene. Okay, now, in this gene there will be one of those two strands that is incredibly important and is actually going to be used by the RNA polymerase II enzyme to produce the piece of mRNA. Okay, the strand that is actually going to be used by RNA polymerase II to actually make a piece of mRNA is known as the coding strand, whilst the other strand, which I won't colour in, is known as the non-coding strand. Okay, right. So what's now going to happen is the RNA polymerase II enzyme is going to work its way along the coding strand, like so, and produce a piece of mRNA that is complementary to that coding strand. So here in blue, that is a piece of mRNA complementary to that coding strand. Okay, now let me pull that piece of mRNA out here. Okay, so here it is. Now, this piece of mRNA, which we have just got by transcribing the coding strand here, this is not ready yet to actually go through a ribosome and be translated into a protein. Okay, instead it is what is known as a piece of pre-mRNA. Okay, it needs to go through what are known as post-transcriptional modifications before it can actually be put through a ribosome and translated into a protein. Okay, now the most important post-transcriptional modification that it needs to undergo is a process known as splicing. Now, what happens in splicing is that certain portions of this pre-mRNA are going to be cut out. Okay, so let's take a step back. The reason that this pre-mRNA is not ready to actually go for a ribosome yet is that certain portions of this pre-mRNA are not actually supposed to be translated. Okay, so the portions that actually are supposed to be translated, I'm now going to box them. Okay, and I don't know actually how many of these you have in the case of COMT, but just to highlight the principle, let's say that you have 3 regions that actually are supposed to be translated. Now, these regions that actually are supposed to be translated, these are known as the exons. Okay, so I'll outline them in orange here. So here's one exon, here's the second exon, and here's the third exon. Okay, now the portions in between the exons, which are the portions that actually are not supposed to be translated, these are known as the introns. Okay, so these are the introns. Now, in splicing, what happens is you cut out the introns and then stitch the exons back together to make a much smaller piece of mRNA known as a mature mRNA. Okay, and I'll colour this one in orange, which is just the 3 exons here sewn together. Okay, and this mature piece of mRNA can now go on and be translated. Now, this process of cutting out the introns and gluing the exons back together, this is known as splicing. Okay, so that's all well and good, but this doesn't explain how you can get multiple splice variants. Okay, right. So, the concept of multiple splice variants is that for some pre-mRNAs from some genes, there is not just one way of splicing the pre-mRNA to get a mature mRNA. Some pre-mRNAs have multiple different ways that you can splice them. Now, let me give you a very nice and easy to understand example of this. Let's say that this final exon here, exon number 3 here, let's say that this isn't compulsory. You don't actually have to have this in your mature mRNA to produce a functional protein. Okay, that would mean that when you were given this pre-mRNA, there were 2 options for how you could splice it. Either you could put in all 3 exons and get a long mature mRNA, or you could do it a different way. You could say, actually, I only want the first 2, and you could then end up with a shorter mature mRNA. Okay, now, if you translate these 2 different mature mRNAs, you will end up with different proteins. This protein will be shorter than this protein. Okay, and those 2 different proteins are now the different splice variants of this gene. Okay, so this is how you can produce 2 different proteins from a single gene. Now, you know, each gene is going to make a huge number of different pre-mRNAs, and some of the pre-mRNAs will be spliced like this, and some of them will be spliced like this, which means that in your cell you're going to get both of these 2 different types of proteins being made for this gene. Okay, right, so that's what's going to happen in the case of the COMT gene. You're going to get 2 different forms being produced. You're going to get what's known as S-COMT, okay, and you're also going to get what's known as MB-COMT. Okay, and I'll go over onto the next page now to talk about these. Okay, so let's start with S-COMT because that's the one we're going to see first. Okay, so S-COMT stands for soluble catechol O-methyltransferase, and I won't rewrite out the catechol O-methyltransferase. Okay, and basically this enzyme is floating around in the cytoplasm, hence why it's called soluble catechol O-methyltransferase. Okay, so let's add it onto our picture here. Now, it's also capable of breaking down dopamine, so we'll put it here. So here it is floating around in the cytoplasm. So this is our soluble catechol-O-methyl-transferase. Okay, and we'll colour it in blue here, and some of the dopamine that's created is going to be destroyed by the catechol-O-methyl-transferase. Okay, so there are these two mechanisms, monoamine oxidase B and soluble catechol-O-methyl-transferase, for destroying the dopamine that's just been produced, and this results in not all of the dopamine actually going into synaptic vesicles. Okay, right, so moving on with this story, what will happen is when an action potential arrives, the synaptic vesicle will fuse with the presynaptic membrane, releasing the dopamine into the synaptic cleft. Now, the dopamine is not just allowed to stay in the synaptic cleft forevermore, okay, because you just build up dopamine up, and it would never stop, okay. You have to then remove the dopamine after it's finished, okay, and what happens is the neuron reuptakes the dopamine back into the axon terminal, okay. So let's now talk about the mechanism for reuptaking dopamine. Okay, so basically there is a transporter that is in the presynaptic membrane which is capable of transporting dopamine back into the cytoplasm. Okay, like so. And this for short is known as the Dopamine Transporter, or the DAT. Okay, so the DA is for dopamine, so the D for dope, and then the A for amine, okay, and then it's the Dopamine Transporter. Okay, right, now this again is a secondary active transporter, okay, so it's going to use ionic gradients to release the energy that is needed to bring the dopamine in. Now, the VMAT2 is what is known as an antiporter, okay, which means that it moved one substrate in one direction and the other substrate in the opposite direction, so they were going in opposite directions, okay. The dopamine transporter is going to be what's known as a symporter, okay, so this is going to be a symporter. Both substrates are going to go in the same direction in the case of the dopamine transporter, okay, which is that they're going to both go into the cell. So what is the ionic substrate for the dopamine transporter? Okay, well, in fact, it is sodium ions and also chloride ions. And just let me close the blind for a second, okay, to try and reduce the amount that the sun affects the... Okay, that's a little bit darker, but never mind. Right, at least we don't have that sort of glare now. Okay, right, so back to the dopamine transporter. So the substrates then for the dopamine transporter are dopamine, and now we want to know the ionic substrate, okay. Well, there's quite a few ions going to be moved by this dopamine transporter, okay. Every time a dopamine moves in, the dopamine transporter is also going to bring 2 sodium ions and also 1 chloride anion. Okay, so let me try and show this. In fact, no, I'm not going to try and show this. So you bring in 2 sodium ions and 1 chloride anion. We don't have enough space really, well, I'll try. Okay, you have 2 sodiums there and 1 chloride as well. I'll put one more arrow, there you go. Okay, which are also going to be moved into the cell every time a dopamine is moved in. Now, sodium is most definitely coming down its electrochemical gradient by being moved into the cell. The sodium concentration extracellularly is around 145 millimolar. Okay, the sodium concentration intracellularly is around 12 millimolar. So that means the concentration gradient is most definitely favouring the movement of sodium into the cell. Now, you also have to consider the electrical gradient, okay, which is why we say the electrochemical gradient rather than just the concentration gradient, okay. Now, the electrical gradient across the cell membrane is usually around negative 65 millivolts. Okay, now that means that the electrical potential intracellularly is 65 millivolts lower than the electrical potential extracellularly, hence there being an electrical potential difference across the cell membrane of negative 65 millivolts. Okay, now, sodium is a positively charged ion, hence It wants to go where the electrical potential is lower, which is the intracellular compartment. It's lower by a whole 65 millivolts. So both the electrical gradient and the concentration gradient for sodium are favouring the movement of sodium in. So most definitely the electrochemical gradient of sodium favours its movement in. Okay, which means that when you move two sodiums into the cell, you're going to be releasing energy. In the case of chloride, it's a little bit more difficult to say because the concentration gradient of chloride generally favours the movement of chloride anions in. Okay, so what I mean by that is chloride concentration extracellularly is generally much larger than it is intracellularly. However, we can't just consider the concentration gradient, we also have to consider the electrical gradient. Okay, the intracellular electrical potential is lower than the extracellular electrical potential by 65 millivolts. Chloride is a negatively charged anion. It doesn't want to come into the place where the electrical potential is lower. It wants to stay where it is higher. It wants to stay outside. So the electrical gradient and the concentration gradient for chloride are pointing in opposite directions, basically. So chloride most likely isn't going to release any energy at all by moving it in. It may even cost a little bit of energy to bring it in. Okay, so the important thing in this case is that you're bringing in the two sodium ions, which are most definitely going down their electrochemical gradient and are therefore releasing energy by doing so. And you're going to use that energy to move the dopamine in. Okay, right. Then once you've brought the dopamine back in, it's then in the cytoplasm again. It can either be destroyed by the soluble COMT or the monoamine oxidase B, or it can be transported back into synaptic vesicles by the vesicular monoamine transporter 2. Okay, finally, I should also mention the membrane-bound COMT. So the membrane-bound COMT is an integral membrane protein, and it's believed to have its enzymatic face facing outside, although this is still controversial. Okay, so it's believed that it is capable of breaking down dopamine that is in the extracellular fluid, basically. Okay, so it will also be removing dopamine from the synaptic cleft. Okay, so that's the membrane-bound COMT. Right then, that's the dopaminergic axon terminal discussed in a bit more detail then now. In the next video, what we will do is we'll discuss the anti-Parkinson drugs, which are all going to affect dopamine levels in the substantia nigra. Okay, sorry, not in the substantia nigra, in the dorsal striatum, trying to bring the dopamine level back up so that we get more dopaminergic transmission, and therefore we have the more ease in initiating voluntary movements.